You have a perfect antioxidant called Catalase. It takes two molecules of hydrogen peroxide, it converts them to water and oxygen, and it doesn't require any other cofactors. It just sits there and does its job. It's amazing. And you don't use it much in your mitochondria, where most of the hydrogen peroxide in your cell is generated. I think that's interesting. The other alternative is you can get rid of hydrogen peroxide using something called glutathione. Glutathione also converts hydrogen peroxide to water, but it has this much more complicated mechanism. It cycles back and forth between reduced and oxidized glutathione. It uses two enzymes called glutathione peroxidase and glutathione reductase. Glutathione reductase is regenerated by NADPH, which in turn is regenerated by the NADH from your food, which comes up here in an NNT, an enzyme called NNT. The NADH from NAD is swapped out to make NADPH, and the NADH becomes NAD+. And so this glutathione is converting NADH to NAD+, and Catalase is not. Glutathione is getting you out of reductive stress. It's actually keeping your NAD pool oxidized, and Catalase is not. That's the fundamental difference between using Catalase and glutathione. And I wondered if there were any examples in the literature of using Catalase and instead of glutathione, and what the effects of that might be. And I found a very interesting paper. So this paper is called Enhanced Expression of Catalase in Mitochondria Modulates NF-kappa beta Dependent Lung Inflammation Through Alteration of Metabolic Activity in Macrophages. So in this experiment, what they did was they took mice and they actually took the enzyme Catalase and they overexpressed it in the mitochondria of those mice. So now what happens is we're shifting the balance of detoxification of hydrogen, all going through glutathione or thioredoxin, which acts much like glutathione. A lot of it is being eliminated by Catalase. And Catalase, as we've said, is not oxidizing NADH back to NAD+. And so you might think that Catalase, having a lot of Catalase in the mitochondria, would actually increase the NADH to NAD+ level. And that's exactly what they found in this paper. They took the mice and they injected into their lungs something called LPS from E. coli. LPS is lipopolysaccharide, and it's something that invokes an immune response. Let's say your lung cells see that lipopolysaccharide and they think, okay, we're being attacked by E. coli, we have to generate a response. And one of the things that cells do to fight off bacteria is they generate a lot of superoxide and they generate a lot of reactive oxygen species. And that actually kills the E. coli or helps beat the E. coli. And so what you saw is that in mice, where they were overexpressing this Catalase to get rid of the hydrogen peroxide, they actually did succeed. The Catalase did its job. It got rid of a lot of the hydrogen peroxide, especially when challenged with this LPS. However, it made the inflammation in those cells much worse. We have an ancient transcription factor called NF-kappa beta. It means nuclear factor kappa beta. It is the oldest known transcription factor that's involved in controlling inflammation, that's involved in controlling our immune response. It's something like 800 million years old. For a long time, we thought that oxidative stress activated NF-kappa beta and caused inflammation. There's a paper about this, there's a review paper about this from 1991 or 1992 talking about exactly that fact, that oxidative stress is, causing this inflammation. This paper refutes that. This paper shows that due to the extra catalase in the mitochondria, the cells with the extra catalase have lower levels of hydrogen peroxide. The catalase is absolutely doing its job of eliminating the hydrogen peroxide, eliminating the oxidative stress, and yet there is more inflammation. And that's because the catalase causes a rise in NADH levels, because catalase isn't glutathione and it's not oxidizing the NADH pool. It's allowing the NADH to build up. And guess what? NF-kappa beta is turned on by acetylation. When NADH levels build up, metabolism slows down, acetyl-CoA levels rise, enzymes become acetylated. There's an enzyme called sirtuins, or SIRT1, that removes the acetyl groups, but that sirtuin is blocked by NADH. And so when we see a rise in NADH and acetyl-CoA together, that's reductive stress, and it causes both oxidative stress and also, it turns out, inflammation, because the acetyl-CoA with high levels of NADH results in acetylated NF-kappa beta. And so reductive stress is the thing causing both the oxidative stress and the inflammation via the acetylation of NF-kappa beta. To confirm in this study that NF-kappa beta is actually turned on by reductive stress, the authors took some of the cells and they incubated them in the presence of lactate. And lactate increases the NADH to NAD plus ratio. Lactate increases reductive stress. And in fact, those cells that were incubated in lactate have more inflammation, more IL-6, more of all kinds of markers of inflammation than the cells that are not incubated in lactate. They did a similar thing that actually used a pharmaceutical that reduces reductive stress, that increases the NAD plus to NADH ratio and oxidizes the NADH pool. And those cells showed less reductive stress and less inflammatory response. All of these signs are pointing to the real thing that causes inflammation via NF-kappa beta is reductive stress, and the way that that happens is through acetylation of NF-kappa beta. I guess if there's a take-home message, it's don't mess with the antioxidant system that you have. You probably want to use the antioxidants that you evolved to use in the places that you evolved to use them. Can you get out of reductive stress? You can. I've been recommending a pair of supplements. Calcium pyruvate eliminates NADH from cells directly, and L-carnitine helps you eliminate acetyl-CoA from your mitochondria. These are the keys to getting out of reductive stress. You can save 12% off both products at bulksupplements.com if you use my promo code FIRE. That's FIRE. Save 12%, get out of reductive stress. And I'm going to have a cool video in the next week showing exactly how I use these supplements because there's been a lot of questions. Thanks for watching.